Dr. Miguel Jimenez earned his BA in chemistry and chemical biology at Harvard University and his PhD in chemistry at Columbia University. He's a postdoctoral fellow at the Laboratory of Langer, working on the development of microbial devices for therapeutics and diagnostic in low research settings, and part of the Academy of Bioastronautics at the Translational Research Institute for Space Health. Dr. Jimenez, we are looking forward for your speech.
So first of all, thank you everybody for having me here. And I'd like to special thanks to PharmaSpace and GPSF for giving me this opportunity. I'm very excited about all the talks that have been happening today. Okay, so today I want to tell everybody about how we're imagining a future that involves a pharmacy that's made out of microorganisms, and specifically looking at how microbial devices can make that future a possibility, and how we're imagining that they will be able to be applied to space therapeutics and space health. And so, as Effie said very briefly, my work really focuses on looking at how we can think of diagnostics and therapeutics, but with a specific focus in areas that have limited resources, both on Earth and in space. Just a brief background about myself. So as you heard, my initial training is a synthetic chemist, but I very quickly got very excited about the idea of using microorganisms as either reagents, tools, or actual products. So then I focused a lot of my work during my PhD on developing ultra low cost diagnostic devices made out of yeast. And since then at MIT, I've really focused on really thinking about the challenges that we have to overcome to take really cool technologies like microbial therapeutics and be able to actually use them as products. And so what we've come up with is this idea of putting microorganisms into devices to actually make that happen. So to sort of get started, again, I think in this setting, you are well aware of the challenges that are affected by space travel. And so what I love to think about is that really the challenges of space are really the same challenges that we face on Earth, but they're just more compounded, even more complex. Specifically, we like to think of low resource settings. So the low resource setting of long duration space travel really makes a lot of the challenges in low resource settings on Earth even worse. So specifically what we think about are three things. Drug stability, as you just heard in the previous talk, is a big challenge. So on Earth, for example, there's many places around the world where you simply cannot have a cold chain, where you cannot keep a medicine cold or ultra cold for the entire transition from manufacturing to actually dosing a patient. And the same goes for space, but even more so, you have very limited power in space. The other main challenge that we face is patient adherence here on Earth. So there's many treatments, for example, like tuberculosis, antibiotics that are a cure for a disease, but because of the extreme pill burden that is required, so people need to take multiple pills for multiple months at a time, you still are not curing some diseases. And so the same thing happens in space, where you have very limited personnel time because astronauts or other civilians will need to be focused on other activities. The final one is the idea of a limited pharmacy. So on Earth, in certain settings, or many settings actually, you have a minimal health system. And in space, you have the same thing where you have limited storage. You have this huge strategic challenge because you need to decide which medicines to take with you before you know what diseases you might need to treat. And so this is a really restrictive and is a big risk to long duration space travel. Okay, so and so what we've been thinking about in our group is, can microbial devices overcome all of these challenges, right? So rather than trying to improve on sort of current medicines, we're really trying to see, okay, is there a new way of doing all of this? And so you might ask, what is a microbial device? And so a microbial device is basically, so in the group of Robert Langer and Giovanni Traverso, they've been developing what we call gastric or GI resident devices. So these are devices that have the form factor of a small pill that can be ingested orally. When the gelatin capsule dissolves in the stomach, it can pop open and change its shape and size, and therefore it becomes stuck in the stomach. And then it can be used as a platform because it stays in the stomach over even multiple weeks, and we're now working on multiple months of residency. And so what my work has been is trying to leverage some of these platforms and combine them with microorganisms to be able to have microbial therapeutics that actually might be able to work in a limited resource setting. So let me give you a little bit more details about these gastric resident devices. So as I mentioned, these devices change shape. So basically you have an elastomeric core, which is flexible, and then stiff arms. And so these STAR devices can be folded up into the shape of a small pill. And again, once ingested, it pops open, and then they remain stuck or resident in the gastric environment. And then for safety reasons, we actually, the group has developed these pH sensitive linkers and other approaches so that after a certain amount of time, the actual device falls apart. So the arms disassemble, and then the whole device at a predetermined time is able to pass safely and transit through the rest of the GI tract after it's released its medicine. And so these devices have now gone through various versions. And so we're able to actually just formulate a lot of different medicines. And so for example, each one of those arms on a STAR device can have a different medicine, and not just a different medicine, but also a different formulation. So for example, you can have a formulation that is fast release and then another one that's slow release. So you're able to target multiple windows of dosing. To make that actually work with microorganisms, what I've been working on is making microbial formulations. So can we mix microbes in a dry state, potentially with polymers, and put them into these devices? And so what these devices have shown though, with traditional drugs is, for example, that rather than taking a single pill and having it only release drug on day number 1, we can actually have these sustained release devices where you're only administering the device one time orally, but then we're able to see release of the drug over a week or more of time. And so these have now been shown, as I mentioned, with traditional medicines. So HIV, antiretroviral, small molecules, anti-malarial drugs, as well as contraceptive drugs to make once weekly administration versions of all of these cases. Now you know a little bit about what gastric resident devices are. And so let me tell you a little bit more about what we're doing with microorganisms to get this to actually work out. And so you might ask, why microorganisms and why can they even be used as therapeutics? And so maybe less of you know this, but actually there's been a lot of interest and push to actually make drugs where the active pharmaceutical ingredient, the API, is a living microorganism. And so a lot of that work has been mostly, 10 years ago, was mostly just in the laboratory, but actually now there's several commercial enterprises here listed on the right side. So as you can see, 10, 14, 15, there's new companies almost every year or every year that are really starting to look at this problem of how do we make drugs out of microorganisms. And so here on the left side, I've plotted, these are the number of clinical trials that are run by companies that are trying to actually develop a product where, again, the product is a living microorganism. As you can see, in the last couple of years, there's been an uptick in the number of clinical trials and really getting quite advanced. So we don't yet have an approved therapeutic that is a living organism, but we're getting very close. So probably within the next 5 years, I would say, there will be an approved therapeutic that is a living organism. And here I like to pause and distinguish this from probiotics, which are things that you can already buy commercially. However, these ones are not probiotics. I would call them microbial therapeutics, where they've actually been designed or specifically studied to target the mechanism of a disease. And again, that's why they're being run through clinical trials to be approved as just a drug rather than a food supplement. So what kind of diseases or what kind of therapies can be made from microorganisms? And actually, the range of therapies that can be targeted is actually quite wide. And so obviously, a lot of the initial work has been done on infectious diseases, because you can imagine there's a lot, because of your microbiome, microorganisms can be used to modulate the populations of microbes in your gut. And so C. difficile, which causes a diarrheal disease, has been one of the great successes of using microorganisms as a therapy. And so several of the companies are actually focusing on using microbes to treat C. difficile infections. But it's very interesting because if you look at the whole range of things that the industry is targeting, again, microbial therapeutic industry, we see immune disorders, cancers being treated, metabolic diseases, and even autism spectrum disorders, because there's even possibility of treating neurodevelopmental diseases with microorganisms. So really the field is quite wide. And I personally believe that this could be sort of the next generation of therapeutics. However, even though there's been these clinical trials that are starting to happen, translation out of the lab still remains a major challenge. And so we've identified two specific challenges. And so one is delivery. So in the sense of drug delivery, unlike a small molecule drug, bacteria or other organisms are alive, and they need to stay alive to actually carry out their function after being ingested. And so in general, they're more on the fragiles. They're fragile. And so they have very low viability when freeze-dried. And also the other aspect of drug delivery is that once you ingest them, even though there's a possibility that they can remain in your gut, what's actually been seen is that they actually have a short-lived effect when you try to engineer them to target a specific disease. So you ingest a little bit of them and they actually get cleared from your system very quickly. So these are two major challenges to actually get it to work in patients. And the second one is a little bit more future forward, and it's that the complexity around using microbial therapeutics is very hard. So a lot of the examples that have been demonstrated in the literature have been in a lab environment, but this is entirely different than the environment that might be faced during the manufacturing process, the ingestion process, and then the actual process of the therapeutic being in your intestinal tract. And so these non-lab environments are very hard to mimic in vitro. And therefore, a lot of the things that have been developed in the lab are having a hard time translating into action, into an actual product. So these are the two major challenges to making this a reality. You might ask, for example, for probiotics, I don't know if they're very common in Greece, but at least in the US, what you can do is you can just go to a pharmacy or even just a supermarket and buy bottles of probiotics, which are supposedly microorganisms that you can just take. But we've actually done a kind of a deep survey of probiotics and found that most of the commercial products actually have extremely low viability, or viabilities that are not enough compared to the viabilities that are needed in the clinical trials that are being carried out by these companies. How are these companies actually carrying out these clinical trials? Rather than using powder-filled capsules, what people are doing now is actually just using, I guess, more extreme methods to actually be able to measure an effect in these clinical trials without yet solving the problem of drug delivery. So one of the two major approaches that are being carried out now are rectal enemas. So this would be, for example, for fecal microbiota transplants. They're usually administered either rectally as a solution or even nasointestinally, again, as a solution. And so you might imagine that for a patient, and if we think specifically for a patient in space, this is not really a viable option to do. The other option that's been looked at is actually making big, dense slurries or kind of milkshakes of bacteria that need to be frozen at ultra low temperatures to maintain viability. So this is okay for now because you can still run the clinical trials, but it is a looming challenge to the field to actually get some of these things to be useful. And in particular, if you want them to be useful in low resource settings, such as long duration exploration space travel. So my focus has been in developing high throughput screening platforms to actually look at hundreds of materials combined with lots of different model organisms. And so we're able to culture the organisms, mix them with a library of materials. Usually we're focusing on GRAS ingredients, which stands for generally regarded as safe. So these are ingredients that you might be familiar in the pharmaceutical manufacturing industry, but actually combining them instead of with small molecules, combining them with these living organisms. So we're able to culture them and combine them in a high throughput manner. We've been able to screen, as I mentioned, hundreds of materials and conditions, and we've actually found hits. So these are like specific ingredients that demonstrate the maintenance of very high viability of these organisms. So we've now been able to validate some of these hits and have shown, at least with gram-negative bacteria, that some of these hits are able to maintain a significant level of viability for longer than 1 month at room temperature. So this involves no refrigeration and no other method of stabilization except for this ingredient that we have found. And so we think this is really an amazing sort of discovery that really opens up the door of using traditional pharmaceutical manufacturing techniques, but now applied living microorganisms. So we've now done that, and we've used these stabilized microbial powders to actually demonstrate that we're able to press tablets. So instead of like just a gelatin capsule filled with a loose powder, we're actually able to press them into capsules. And again, looking at the traditional pharmaceutical manufacturing techniques and show that the viability is maintained throughout those processes. And as I mentioned, the reason we're excited about that is because now that we can formulate microorganisms in this traditional way, we're now able to directly combine them with these microbial devices. So we think that we've made a big headway in the delivery challenge. But then, as I mentioned, there's also this complexity challenge. So let me just very briefly finish up by explaining what I mean here. And so what we can do is we can actually look again to those clinical trials as an example and compare them to what people have done in terms of genetic engineering, microbial therapeutics, comparing clinical trials versus the academic examples that have been done in laboratory environments. And so when we compare those, actually the clinical trials are all using very simple standardized microorganisms. So with very few or very simple genetic modifications, and most of the time, even no modifications. So a lot of people are just looking to discover new microorganisms, but not really engineer them that much. However, in the academic literature, there's a huge field of synthetic biology that has made really incredible complex systems that can sense and respond to different stimuli and, for example, send out signals or different therapies. But these are not the ones we're seeing in clinical trials. And the reason is because complexity is hard. And some of the parts and examples that have worked in the lab end up not really working in clinical trials. And to solve this challenge, what we're working on currently is actually developing microbial devices, not just as a therapeutic product, but also as a discovery platform. So being able to do in situ genetic profiling of the microorganisms in these devices is allowing us to generate a new set of parts, genetic parts, that will be robust and actually function as expected in the context, in the target environment. So with that, let me finish. And I just want to highlight that while we're doing all these things with thinking of space travel as our frontier of challenge, what we actually hope for is that all these technologies that we're working on will be just as useful here on Earth and solve some of these major challenges like no cold chain, pill burden, and a minimal health system. So with that, I'd like to thank a ton of people that have worked with me to make this a possibility, and special thanks to PharmaSpace and the Greek Pharmaceutical Student Federation. And please, if anybody wants to get in touch, I think a good way would be on Twitter. And so here's my Twitter handle, jmigueljd. I'd love to take some of your questions.